I'm Allison Jonjak, the Cranberry Outreach Specialist with the University of Wisconsin-Madison Division of Extension. These videos are the on-demand recordings of UW Extension and the Wisconsin State Cranberry Growers Association's virtual brown bag lunch series. These monthly sessions offer growers and researchers a way to share timely and relevant information. We hope you'll enjoy and continue to engage with us.
Hi, everyone. My name is Grant Holly. I'm the Executive Director of the Wisconsin State Cranberry Growers Association. We are very happy to be able to partner with UW Extension to bring you the research needed to be able to navigate our ever-changing environment. I'd like to thank all of our speakers today for their valuable time and leadership, the growers for joining us, and Allison for being the catalyst from which all of these sessions rely. Thank you so much.
So for a little bit of housekeeping, I have captions that are enabled that you can turn on if you don't see them, if you want to be able to follow along and prefer a transcript format. If you have questions throughout the presentation, you're welcome either to unmute yourself and ask them or to use the chat window, and I'll be following the chat, and then I can ask the questions on your behalf. If you would like to ask the question publicly, you can use the drop down to everyone. If you'd like to ask the question anonymously, use the drop down and choose to Allison Jonjak, and I'll ask it anonymously on your behalf. Amaya, I'll let you say hello as well before we kick things off.
Sure. Hi, everybody. I'm here. I'll be here for the end of the presentation that I gave in July, and I'm happy to answer any questions and discuss anything that people want to discuss about the topic.
This many times is so hard to do. This work is so time consuming and so intensive that we just can't do it for all the cultivars. And in this kind of study, we would assume, because it's mostly anatomy, that we have a lot of similarities across the cultivars. There might be some slight difference, but a lot of what I'm talking about today, I think, is generalizable to all of the cultivars. We did this work at the Cranberry Research Station, and we're very thankful for Wade and Max for always facilitating and helping us, and also Beth in her role there for helping us to do this kind of work there. And we did the study in 2 years, in 2022 and 2023. And so let me show you a little bit of some of the results that we got. So the first thing we looked into was just, general fruit development. What happens with the fruit? How does the fruit grow? And so what you can see here on the right axis, we have fresh weight of berries, and then in grams, and then on the axis on the right side, we have the dry weight. Okay, so we took that fruit, we dry it completely, and then we took that weight. And what you can see here is like we have an increase, obviously, in fruit size, a little bit more steep at the very beginning, and then it kind of like plateaus as we are approaching harvest, which is what we know. And then the dry weight sort of like follows a very similar trend. It's just like lower values, because a lot of these is basically, it's just water in the tissue. So there's you can see they start with very little water. It's just mostly dry matter, and then as the fruit develops, there's quite a bit of water that accumulates there. And I have some pictures here of what does it mean, like July 15, where were fruits there? Well, about like a little bit pea size, a little bit smaller than that. Well, August 15, you have fruits are almost fully developed, and we start to see some flush, and then here by mid-September, early September is when we harvest. So this is just fruit development, how fruit are gaining weight, are gaining the growing. And here's the curve. As we were doing these things, here's the curve for potassium. And so here you can see the accumulation of potassium in the fruit. And so very similar to the weight and the fresh weight of the berries, you can see that there's quite a bit of increase in the absorption and the accumulation of potassium in the fruit at the very beginning. Whoops, sorry. Oh my God, I'm messing it up, sorry. There's quite a bit of gain here from July 11 all the way to about like August 15 of potassium accumulating in the fruit. But then you can see very similar that at the very end, you can see sort of this like plateau. There's no more potassium accumulating in the fruit. And potassium is one of those nutrients that is accumulating in the fruit through the water, as water is getting in the fruit driven by transpiration, a lot of that potassium is also getting in the fruit. And so, you know, not all nutrients get in the fruit in the same way, but this is for potassium. And I just put this line here, just showing you like this different, like this kind of like break in which we have like quite a bit of accumulation at the very beginning. And then again, we see sort of at the very end of the season, this plateau, and that no more potassium seems to be getting in the fruit by that time. Here, there's a bunch of different nutrients. So very similar to potassium, it's just this graph contains calcium in black, magnesium in orange, and then phosphorus in green. And so what you can see here is, again, we see that same trend for magnesium and for calcium, that we have, you know, an increase in the accumulation at the very beginning of fruit development, but then that just plateau and the concentration is the same. In the case of phosphorus, you see that there's a little bit, you know, it continues to increase the amount of phosphorus in the fruit. And one of the reasons for this is that phosphorus is also, you know, linked to many sugars. So you can get phosphorus accumulation into the fruit just because carbohydrates are still accumulating into the fruit. And those are not getting into the fruit through the xylem, but through the phloem. So this is the other set of pipes that are, you know, in those pedicels that are bringing the sugars from the leaves through photosynthesis into the fruit. So these are two different, you know, highways, one that is bringing just the water and another one the sugars. So in the case of phosphorus, it seems that we have maybe at the very beginning of the season, there might be some phosphorus that is coming from the soil through water and the xylem, and then maybe in this other end of the season is coming through sugars and through the phloem. Okay. So again, we still see this kind of timing in which the fruits are about almost full size, and we're starting to develop that. We're starting to see that change in color from green to sort of like yellowish, and we start to see the first blush. That's when we start seeing that a lot of nutrients, such as like the potassium, the calcium, and also in this case, the magnesium, seem to be, you know, there seems to be no more accumulation in the fruit itself. So one interesting nutrient that had a completely different type of curve when we were looking at the accumulation in the fruit was the boron. And so we have this, you know, bell shape on boron that's very different, and we try to figure out, okay, what would this be happening with boron? And boron is a micronutrient. The plants need it in very low quantity, but it's an essential nutrient. It's very important for pollination. So maybe not incredibly important for in sense of like, you know, fruit development, though it does play an important role when it comes to cell wall and firmness, because a lot of the boron, some of the boron is associated with some of the pectins and the hemicellulose that we have in the cell wall that gives a structure to that pulp. So if we have deficiency of boron, we can also have, you know, softer fruit. So in a way, it's important for fruit quality. But what we see in this case is that we see like a steady increase, and then it just really drops the concentration, and then it kind of like plateaus at the very end. So boron, we don't know why is that. We hypothesize and we think that a lot of what's happening here is that some of the, you know, that boron is immobilized and remobilized and moved from the fruit in and out of the fruit. But this is one of those nutrients that have sort of like a different pattern, and we really haven't explored really well why this is happening, but just to show you that not all nutrients behave the same. But the major micronutrients seems to, you know, follow that trend that I just explained, in which we increase and then we tend to plateau in mid-season. So as I was saying before, a lot of these nutrients move through the xylem and move through transpiration. So one of the things that we wanted to look into was like, well, is there something maybe happening in the pedicel itself? So is there something happening here as the fruit develops that might be affecting the movement or conditioning the movement of water and nutrient into the fruit? And so besides just looking at the amount of nutrients that we found in the berries, we also did some sampling of those pedicels as the fruit developed, and we did some microscopy work, and we wanted to really look at what is happening with these vessels. And I call them like pipes because that's what it is, that they're moving this water and nutrient. Is there maybe something like they're stopped functioning? What do we know about it? And is there something that it can tell us about nutrient movement into the fruit that could inform us about how to change some of our fertilization practices? So what I want to show you with this graph is showing the xylem vessel density. So how many of those vessels do we have in those pedicels as the fruit develops? So when we started, here at this size, fruits were very, very small. Actually, we started at pinhead. And we would take those pedicels, we would make cuts on them and look in the microscope and try to figure out what's the density of the xylem vessels that are moving the water and the nutrient into the fruit. And what we find out is that as the fruit develop, we see an increase in the density. So we see more and more and more of those vessels being developed. But then again, at this kind of critical stage in which we start seeing color development and this change between the green fruit to kind of more like yellowish fruit and then the blush development, we started seeing that this sort of like plateau, and here we see sort of a bit of a decline. So the density increased, but then again, the same trend sort of plateau. But then when we looked at the hydraulic mean diameter, this basically is trying to calculate and estimate what is like the overall like potential of moving water. It's not only the density and the number of those vessels, but what is their potential of how much water they can actually move through the pedicel. We see that as the fruit develops, there's the hydraulic diameter, the amount of diameter they have to move water starts decreasing. There's less and less water that can actually move through the pedicel into the fruit. Okay, so that was something really interesting. It seems like the flow of water is much, much bigger at the very beginning in fruit development, and then it really kind of like decreases as the fruit grows, and it steadily decreases all the way until harvest. So less and less water is getting in, is moving from the pedicel into the fruit. And so we're like, oh, well, should we also start looking at whether these are all anatomical like measurements. So like we knew that there were higher density of vessels, but that overall those vessels were getting smaller and smaller and smaller. So like overall the capacity of moving water was less as the fruit was getting ready to harvest. But another thing that we really wanted to look into is like, well, are they really functional? Are they actually moving any water and nutrients into the fruit? Because we saw that plateau, and so that means like, well, maybe there's like nothing happening. That maybe that pedicel is just not functional anymore when it comes to the xylem, so the vessels that are moving water and nutrients. So what Pedro did, which is very smart, is he decided to use, what if we use a dye to figure out by tracking that color whether the water is getting into the fruit as it develops. And so what he did is he sampled a bunch of uprights with a bunch of fruit either attached to the pedicels, as you can see there, or he also detached them, as you can see in the picture in the bottom, and he put them in a little container that had red dye. And we put them into conditions in which you would have a lot of wind that would mimic transpiration. So we tried to create that water vapor deficiency so that it would simulate exactly how the plants are in the field, in which the air is drier, so it's pushing that water out of the leaves. So we try to simulate that in the lab by putting these little uprights in this red dye solution, and then also created the conditions so the leaves and the fruit will continue to transpire and try to move water into the fruit. So here's a really good example. You can see here, we did this for 30 minutes, and you can see at the very beginning when we put the fruit and the uprights in the little jars, and then what happened after 30 minutes, you can see when the fruit is transpiring and the leaves are transpiring, the dye is moving into the fruit, and you can see how everything turned red. So we did this experiment when the fruit started like very, very tiny at pinhead, all the way until we harvest. And so what is it that we saw? Well, you can see here in this graph is the percent of fruit that had dye inside. And so we dissected the fruit to look at that. And what we saw is that at the very beginning, all of the fruit was actually getting this red dye inside. But as the fruit developed, and again, I want to show this image of this critical change of color that is happening, this stage in the fruit development, we started seeing that there was less of that dye, and that dye was only getting at the very at the portion of the fruit that is attached to the pedicel. But then as the fruit continued to develop, none of that dye was moving into the fruit, meaning there was no water and nutrients getting at all into the fruit. And so this was like really, really interesting that we figure out that at some point during fruit development, water and nutrients are just not getting into the fruit anymore. And the critical point seems to be when the fruit reached this, considerable fruit size, but we mostly figure out that it's when the fruit are changing color. So changing from green to this kind of like more whitish, yellowish, and it's the very beginning of the blush and the color developing in the fruit. At that point, when we were analyzing, when I show you those graphs of calcium and of magnesium and of potassium, that's when we started seeing the plateau. And the plateau seems to happen because that xylem stops being functional. And we prove that by showing you that there's no more dye getting into the fruit. And so that shows, it's a really good proof that, water and nutrients are not getting in there. And so here, this curve shows at the stage in which 50% of the fruit will not have dye in any of the portions of the fruit, okay? And so another very interesting thing that we saw as we were doing this test is that we started seeing an abscission zone develop. And so what I mean with an abscission zone, the pedicel started, there was something that was started sort of to detach it from the fruit. And even though we need to really pull those berries to harvest them, there is some sort of separation already happening that might be contributing to the fact that water and nutrients are not getting into the fruit. And this happened in August, and we did a lot of microscopy work, but I think that one of the things that is most evident is that you can see that some of that dye is leaking out of the fruit right here. And so that also shows us that there's some abscission zone that is happening. This is very common on other fruits. We tend not to think about this in cranberries, but if you think like, about like apples and peaches and all that, if you let them ripen the fruit, they will just drop because this is that abscission zone that is happening here. In the case of cranberries, we need to pull the berries out of the vines, but that doesn't mean that there's no abscission zone. And we did show this with this when we looked at this phenomenon of this dye kind of like coming out of the fruit, even though the pedicels were intact. Internally, when we did the cut, we could see that these abscission zones are happening here, and the tissues seem to be rupturing there. So very preliminary conclusions. We have a lot more. I mean, Pedro has a lot more that he has done, and will be, providing more information about this. But it seems that water, nutrient, and sugar movement into the fruit definitely declines as the fruit develops. Okay, and that's especially true with the data that we show with the water and the nutrients. By the second week of August, about 50% of the fruit did not take any dye, which means 50% of the fruit that we tested, there was no water moving into the fruit whatsoever. And then also there seems to be some evidence that there's some fruit isolation suggesting that the water and nutrient movement into the fruit stops approximately about 4 weeks before harvest. So that are some of the preliminary conclusions that we got out of this study. And again, just to acknowledge and thank the funding that we had for this study that has a lot more information in it. So the USDA funding that we got, and also really to thank the station for facilitating the research there and for really helping us do all of this crazy stuff that we do at the station. And so we really want to thank Wade and Max and Beth in her role as the research manager of the station for letting us do this work. And so with that, I hope I didn't go over time, and I'll be happy to answer any questions.
Yeah, we are good to have a couple of questions.
And I actually want to check whether people have questions live today. We can prioritize those. Otherwise, I can start playing through the questions that are on the video, but I wanted to check first whether folks had questions from this watch through. And I've got the chat up in front of me so that you can ask questions as they arise.
If people want to ask their questions either by asking and unmuting and asking. In the meantime, I just made the note for people following along on the closed captions. Closed captions loves to hear pedestal. It is actually the pedicel, Pedicel. If you are if you were baffled by that, I made a
also
note.
I know that my accent doesn't help, but I mean you folks are used to it. So.
No, the closed caption doesn't have a lot of botany knowledge. When I make these updates, there's a lot of botany knowledge that, like, well, it's a good thing we know what's going on. I had made the question and you basically answered it, but in your chart of, for example, boron in milligrams per gram of dry weight means the dry weight of fruit only or tissue plus fruit. That meant the fruit only,
fruit,
right?
Only fruit.
Yes.
Only
okay.
Fruit. Yeah.
Another question. We've got a lot of, wow, very good information and great topic by Amaya. One somewhat related topic, is there any research planned or interest in developing an evapotranspiration model for cranberries, especially as more and more growers get weather stations and could be gathering data? Any thoughts on that?
No, not that I know of, but I know that there's a new researcher here at UW-Madison, and she works a lot on irrigation, and we connected, and I told her, like, we have a lot of things to do on cranberries, so I'm going to try to bring her in, and she's a specialist on irrigation, and so it would be great to have her, to talk to her and to maybe just, I've been already bouncing some ideas with her, but I think that would be a great topic. The only thing that I want to make the distinction, just because you brought this up, when you think about evapotranspiration and all of that, we're thinking about foliage and leaves. What I presented here, and I will be presenting more of this project, is mostly related with the fruit. How's the fruit transpiring? And I think that maybe Pedro talked about this a couple of years ago, but we have some data on there, because it's the transpiration that is moving the water. It's just like, basically like when we drink out of a straw. So the end of the straw is that pull from the environment, from the air. And that's what's bringing the water into the leaves, but also into the fruit. But a lot of the data that I presented today is like related to the fruit itself, which is as a very different organ when it comes to transpiration than the leaves, with that their main goal is to transpire, which is not the case of the fruit.
Absolutely.
We actually did have a question come in just from today's session. Amaya, can high boron levels in tissue samples be a problem? Are there negative effects? And answer quickly, yes, boron can be highly toxic and produce tissue damage. I don't know if you want to add more to that verbally.
Yeah, I mean, boron, it's an interesting nutrient in which, like, the plants need very little of it, but as soon as you get over that range, it can become very toxic, and so there can be a lot of problems with that tissue damage. It looks almost like salt damage. You also have like damage to the roots. So elevated or very, very high levels of boron can definitely be a problem. And so if that's the case, if you have results of tissue analysis with high boron, I would highly encourage you to look into your fertilization record and just try to track back what products are you using that might have high boron. Also, boron is one of those nutrients that tend to be highly available in acidic soils, which is obviously the soils in which we grow cranberry. So that might make it highly available, and also with high organic matter.
Awesome. I will put us back in video mode unless there are more questions here then. Oh, okay. I guess then a question, I presume from Quebec there, but what is high boron ppm in foliage for you? At what point is it critical?
I don't know it from the top of my head, but if you give me a couple of minutes, I looked it up and put it in the chat.
Okay, let's do that. I'll have the video play a little more while you check that, because I also would have to look it up.
And I think we're, I guess I've got a couple more questions already here and probably time for 2 more after these 2. So if people have questions, go ahead and bring them in. But question, so are the most important nutrients nitrogen, potassium, and the micronutrient boron in terms of filling fruit?
So there's the fruit is going to have all of the nutrients, okay? But when we think about like macronutrients, those are the nutrients that we talk about extensively about this, the plant needs in really high quantities. That's what they call macronutrients. So we tend to focus on that just because there's more of those components in the fruit itself, okay? And that's what we're looking at. And the reality is that this study started with the idea of trying to look at calcium. We were mostly interested in calcium because as you all know, my lab has been working a lot in trying to understand like fruit quality and what makes a good fruit to be a sweet and dry cranberry. And calcium is really important for the cell walls and the structure of the pulp of the fruit. And so we were specifically looking at calcium, but since we were doing these, we did analysis for other nutrients. And so we started to figure out that the pattern was very similar. And so boron is an exception. And I show it there just because, you know, it's a different kind of pattern of movement into the fruit. But boron is also, you know, the fruit and the plants really needs a very, very, very small amount of boron, okay? It's one of those nutrients that you go from deficiency to toxicity in like nothing, like very, very, very small range. So it's an anomaly in the way that it moves into the fruit, but also it's not something that we are constantly fertilizing for. What I think is important, and one of the takeaways for me here is that, and I'm sorry if I sound like a pain, because you've all heard me say this so many times about, like, you know, over fertilizing and thinking that fertilizer is going to solve your problem of fruit size, and that we're going to fertilize, we're going to put more potassium because we need to get, you know, because that's going to help us, you know, with fruit size. I really want to show you with this research that fertilization at the end of the season, not even at the end of the season, we're talking about here like mid-fruit development, that fertilization at that point is really not doing anything for you, because I just show you that that water and those nutrients are not getting into the fruit at that point. And so when we get to the point in which the fruit starts developing, you know, the flush, and they start to change color, it seems to be a critical stage in which a lot of other things are also happening. And one of the things that are happening is that the water movement and the nutrient movement into the fruit is kind of like minimal. And it looks like the last portion, maybe like the last fourth or even the third portion of development of the fruit is there's mostly sugars coming from the phloem. I only show what was coming from the xylem. So there's sugars coming from the phloem that are getting into the fruit and are helping to continue to develop it. But in terms of fertility, that we're thinking we're going to put more nutrients into the fruit, that is really not happening. So when you're doing some of those fertilizations in August and late August, those nutrients are definitely going into the plant, and they're going into the leaves and into the other structures of the plants. But don't fool yourself, they're not going into the fruit, and they're not necessarily going to help you to increase fruit size at that stage. And I know it's very frustrating, and we talked about this because there's so many other factors that are affecting this. But I want to add this piece of information just to let you know that when you are going to do those fertilizations, just keep in mind that those nutrients are not going into your fruit.
And I'm gonna, we are like 1 minute over, but I'm gonna smush two similar questions into one question and let you handle that, and then we'll move along and let you head off to the thesis defense. But what is the take home? Is there a fertilizer application that should go on at a specific time to maximize fruit size? And then the second question that's kind of a follow-up, so should fertilization be finished at the beginning of August? You kind of addressed that with the well, after fruit, maybe there are things going into the tissues, but do you have a suggested specific timing for fertilization to maximize fruit size, and then after that it shouldn't be considered for fruit anymore?
Well, if you look at the graph and like everything that was happening, you could see like at the very beginning of fruit development, everything was just going really fast. So the timing of fertilization, you know, when we're thinking about fruit, it has to be at the very beginning of the fruit development. So when those fruits are setting and they're developing their pinhead all the way until pea size, this is the period of really, if we want to get those nutrients into that fruit, that's the period. I think that if you're just moving now, like, you know, mid-August or after we go, like, I don't want to give specific dates because, you know, we know that we have different timings, but when we start seeing that those fruits are changing that slight change of color, it really means that anything that is going in to the fruit through the xylem is really declining. So there's no point of doing. We can fertilize, we can still fertilize, but just remember those nutrients are going to go into, you know, the roots, the stems, the uprights, the foliage, but not into the fruit. And so that's my recommendation.
Thank you.
All right. That takes us through the portion on nutrient and water uptake into the fruit, and hopefully I can seamlessly shuffle from that back to here. We've got a couple more follow-ups on the boron. Normal was 15 to 6D, 15 to 60 parts per million. So looking at above 60 parts per million, considering it toxic. And indeed seeing that adding small doses of boron following granular application foliar analyses did increase drastically and often above 60 parts per million. So Amaya's response that makes sense. You only need a small quantity of fertilizer to raise the level in tissues. So just some good reminders there on that front. Happy to take more questions on the nutrient and water uptake into fruit discussion that we just heard or had. And then feel free, again, continuing, since I'm the second presenter, I am more than happy to pause what I'm talking about to bounce back and let Amaya have an opportunity to answer more questions. So do keep using that chat if you've got questions to ask.